Hello students, so in this session we will be going to study about the anomalous secondary growth in Bignonia. Here you can see the image of the Bignonia plant. So before that, what do you mean by secondary growth in plants? So the secondary growth of the plant is increase in the stem thickness, and it is due to the activity of the lateral meristems which are absent in the herbaceous plants or herbs. So there are two types of lateral tissues involved in the secondary growth, namely vascular cambium as well as the core cambium. So now, what are anomalous secondary growth? So what do you mean by anomalous or abnormal secondary growth? So before that, so what is secondary growth? Increase in the plant girth due to the vascular cambium or the core cambium, that is termed as the secondary growth. Whereas the development, arrangement and the activity of the vascular cambium in most woody dicotyledons as well as the gymnosperms tend to be very similar. So there are some alternatives or deviations which produce new secondary tissues that do not follow the normal pattern. As a result, the secondary plant structure that are formed are termed as the anomalous. So what do you mean by anomalous? Anomalous means the deviations from the normal or common type or order. The term anomalous secondary growth is given for the deviation and the variation. The pattern of the secondary thickening shows the deviation from the normal type in regarding the abnormal or anomalous secondary growth. So what do you mean by anomalous? Anomalous, it is the any deviations or the variations. Okay. So now coming to the anomalies. So why does this anomalous or the secondary growth occurs in the plant? So there may be abnormal position of the vascular cambium, abnormal behavior of the normal cambium, and also the cambium formation and its activity. Coming to the extrastellar cambium, intraxillary, and also they include the phloem also. And there may be presence of the medullary bundles along with the normal cambial activity, and presence of the cortical bundles along with the normal cambial activity, intraxillary or the intral phloem, and also the secondary growth in monocotyledons. Example is the Dracena, and the scattered vascular bundles in the dicotyledons also shows the abnormal growth. And the absence of the vessels in the xylem and scattered vascular bundles along with the cambial activity in the dicots also include the abnormal growth in the plants. Now coming to the Bignonia. So this Bignonia is a member of the Bignonaceae family. The young stem shows wavy outline with a permanent ridges and furrows. So here you can see the tiers of the stem. So the young stem shows the wavy outline with the permanent ridges and the furrows. The most interesting anatomical features in the Bignonia is the occurrence of the anomalous secondary structure. So it consists of the epidermis, cortex, perivascular region, and vascular bundles, and also the pith region. So the epidermis, so it is uniseriate, cuticularized, and it is wavy in outline. Coming to the cortex region, it is a parenchymatous and bonded internally by the starch sheath layer. And perivascular region, it is scattered patches of the sclerenchymatous cells, particularly under the ridges of the stem. So here you can see the epidermis, cortex as well as the perivascular region. Coming to the main vascular bundles, so in young stems the vascular bundles are arranged in ring, and cambium is normal in the disposition. And also soon as the secondary growth starts, the cambial ring is formed, and then it cuts off different proportions of the xylem and the phloem at the different points. At the 4 points, the formation of the secondary xylem is reduced and that of the secondary phloem correspondingly increased. And as a result of this, there is a structure with a ridged and furrow xylem cylinders is formed. Coming to the pith region, pith is usually parenchymatous. It shows the abnormal cambial activity during the secondary growth in the intrastellar region. And what is the anomalous secondary thickening in the Bignonia? It is due to the abnormal functioning of the cambium. And here the cambium is normal in the disposition and abnormal in functions like the adaptive types. And there may be two types presence of the phloem wedges in the xylem as well as the presence of the fuseroid xylem. Fuseroid xylem means having long narrow crack openings. Okay, so fuseroid means long or narrow cracks. So the first one, that is the presence of the phloem wedges in the xylem. So here the young stems which exhibits this type of the structure when mature are provided with the normal ring of the vascular bundles. So the origin, position and the functions of the cambium ring is normal in the initial stage of the growth, and the cambium function normally producing more secondary xylem towards the inner side and the less secondary phloem to the outer side. But after some times the cambium cut off a different proportion of the xylem and the phloem in different points at 4 diagonal. So you can see here, so there are 4 diagonal point arranged in the cross-shaped order. Formation of forming the secondary growth is reduced and that of the secondary phloem correspondingly increases. So that secondary phloem with the transverse bands of the bast fibers get penetrated within a mass of the xylem in the form of the phloem pocket. Hence, the woody cylinder appears to have 4 longitudinal grooves which become increasingly deeper with the secondary growth. As a result, so this 4 furrows at the equidistant points appear in the xylem extending most of the pith region, and the cambium breaks up into the number of stripes, widest ones occurring the opposite and the 4 projection ridges of the wood and the narrow ones at the base of the grooves. And the cambium is not found on the radial surface. Peculiarly, the structure with the ridged and furrowed xylem cylinder is formed. And now the 4 radial groups of the phloem are united by the medullary rays seen transversing the phloem of the furrows. So now coming to the second one, that is presence of the fuseroid xylem. So fuseroid, what I said, so fuseroid means it is a long xylems, long or narrow cracked xylems or opening of the xylems. So now the fuseroid xylem may only be seen in the fairly old stems. So where you will see this fuseroid xylems? So you will see this fuseroid xylems in the old stems. The first of all, the wedges of the phloem are formed and therefore and thereafter the xylem strands become fuseroid by the deletions and the cell division in woody parenchyma and the pith. So now how you will identify the secondary growth on the bignonia? So there are 4 formations of the 4 furrows of the secondary phloem in the secondary xylem. So there is a presence of a 4 furrow of the secondary phloem in the secondary xylem is due to the abnormal functioning of the cambium which was behaving normally sometimes earlier. At 4 or more places, cambium produces less amount of the secondary xylem towards the inner side and the large amount of the secondary phloem towards the outer side. Thus, 4 wedges of the secondary phloem are formed. They intruded into secondary xylem and so xylem cylinder appears ridged and furrowed. So this was about the anomalous secondary growth in bignonia. I hope you guys understood well. Thank you.